It is 565 million years ago during the Ediacaran period of Earth's history, and you are diving to the extreme depths of the ocean in a state-of-the-art research submersible. Here, in the Ediacaran, you're searching for some of the most mysterious organisms that ever existed. This is the Middle Ediacaran, and the Earth was recovering from an incredible ice age called the Gasquier's glaciation. The entire Earth froze for around 2 million years, from the poles to the equator. Life somehow survived this snowball Earth and made it to the hot tropical climate on the other side. Bacteria, archaea, single-celled eukaryotes, and even tiny sponges and comb jellies had the tools they needed to see themselves through the cold. And the really amazing thing? The creatures that thrived after this glaciation stepped up from single cells and suddenly became large, complex, and diverse. And then, at the end of the period, the Ediacaran biota just disappeared as mysteriously as they arrived. You are sitting in the pilot seat of the submersible, staring out of the large glass dome window. Marine snow drifts down through the cold darkness, illuminated by the spotlights. You shiver. Down in these deep ocean depths, it's cold and silent and pitch black. Ah, look, you've arrived at the sea floor. It all comes into focus, an eerie ecosystem of silent, still creatures, unable to move, just standing there. There are spindle-shaped things lying completely still in patterns that look like colonies. Others look like bunches of leaves gently drifting in the deep water current. And right in front of you, attached to the sea floor like plants, are tall fronds that look like a long leaf embedded in the ground. This is the Ediacaran biota of the Avalon assemblage. The tall leaf things are called Charnia masoni. As you shine the spotlight around, you see them everywhere, from tiny ones smaller than your thumbnail to others more than half a meter tall. They are long, flat, and they have segments that branch out into fronds. They are anchored down and can't move, and they have no mouth, no stomach, and no muscles, and they feed by absorbing dissolved organic carbon and other minerals from the water. When they were first discovered, placing them in any kind of category was so difficult that paleontologists couldn't even decide if they were plants or animals, or maybe even some other kingdom altogether that never survived to the present. Once studies on the rocks concluded they lived in deep water over a kilometre under the sea, it was clear they weren't plants. Plants need light to photosynthesize, and Charnia lived in permanent darkness. Superficially, they look more like modern-day animals called sea pens, which are actually cnidarians like corals or jellyfish. But that's where the comparisons end. Charnia and cnidarians had fundamentally different body plans. Sea pens are bilaterally symmetrical, meaning that both sides are symmetrical mirror images of each other. Charnia, meanwhile, branched out from a central zigzag shape. This glide symmetry body is offset, and no living creature today has this shape. As a result, classifying Charnia was a challenge, even today. It was placed in its very own clade, called the rangeomorphs. Look at those brownish things, the spindle-shaped creatures lying motionless. They are another type of rangeomorph called Fractofusus, and they're some of the most common creatures here. Like Charnia, Fractofusus have bodies with modules that grew out in fractals from a center that ran in a zigzag shape. This means that every module that branches off divides into smaller and smaller versions of the original. The way they are positioned is very interesting. They seem to live in communities where a large Fractofusus is surrounded by smaller and smaller ones. This might be because of the way they reproduce. It's thought that Fractofusus reproduced by sending out runners, like the stolon of a plant, which budded off to create a clone that grew nearby. There are other types of life living in this still and silent community. You spot Haeotia, an organism that is probably in the cnidarian phylum, and like Charnia, Haeotia is sessile and anchored to the sea floor. Each one is about the size of a teacup, but with a square shape and four-fold symmetry that branches out into drifting fibers at each corner. It's thought that these were the first muscle fibers ever to evolve. Another resident of this Ediacaran abyss is Bradgatia lymphadensis, a Rangiomorph filter feeder with 6 fractal fronds that grew out like a cabbage. Today, paleontologists have named over 200 species that lived during the Ediacaran. Life in the Ediacaran has been found in groups called assemblages, giving us a clearer idea of which species lived together and when, what their habitat was like, and how they might have interacted. Charnia and Fractophusis come from a fossil group called the Avalon assemblage, which dates from 575 to 560 million years ago, and they've been discovered in Newfoundland in Canada and central UK. The fact that Ediacaran fauna exists at all was once thought to be impossible. Since the 19th century, paleontologists and geologists observed that the oldest multicellular fossils ever found were in Cambrian rocks, and there were lots of them. But in rocks that were pre-Cambrian, the fossil record seemed to go blank. Multicellular life didn't seem to exist. There were theories that Cambrian animals must have had ancestors of some kind. Cambrian arthropods, mollusks, and annelids can't have come from single-celled bacteria. There must have been an evolutionary stepping stone. These evolutionary stepping stones were being discovered, but the scientific community refused to believe that pre-Cambrian animals existed. In 1872, the Canadian paleontologist Elkanah Billings described disc-like fossils of a new animal called Aspidella, but the paleontology community believed they were just inorganic concretions. In 1946, the Australian geologist Reginald Sprigg was surveying silver and copper mines in the Ediacaran Hills in the Flinders Ranges in South Australia. He discovered small, round imprints in pre-Cambrian rock layers, each one about 10 centimeters across and marked with radial lines, but his findings were also ignored. The first fossil of Charnia masoni was discovered in 1956 by a young Leicestershire student named Tina Negus. When she showed the fossil of what looked like a feather or a leaf to her teacher, they didn't believe it was significant. It wasn't until the next year that another student named Roger Mason discovered another leaf-like fossil in the same forest. This time, after being ignored by their teacher again, they reached out to an academic geologist. Backed by expert opinion, there was no doubt that this leaf-like shape was a pre-Cambrian fossil. Nobody could ignore this momentous find. Interest in pre-Cambrian fossils suddenly took off, and Ediacaran fossils were uncovered across the world in the decades that followed, from Russia to Namibia to Brazil. The Ediacaran period was officially named in 2004, after the Ediacaran Hills, where Sprigg found his fossils of a disc-like animal called Dickinsonia. In the 1960s, a treasure trove of fossils were discovered in Canada's Mistaken Point in the Avalon Peninsula, and this massive rise of biodiversity was called the Avalon Explosion. The Ediacaran period began 635 million years ago at the end of the Marinoan glaciation, another of Earth's snowball Earth periods. As the Earth warmed and the ice melted, sediments and nutrients were released from ice caps and poured into the oceans, feeding vital nutrients to phytoplankton and boosting oxygen levels in the sea. You are standing on a red, rocky coastline jutting out into the Panthalassic Ocean. It is 555 million years ago, and the world of Charnia and Fractophusis had already existed for 10 million years in total silence in the ocean abyss. You take out a map of Ediacaran Earth. The continents were very different to those of the modern day, and most of them had accreted together into a huge supercontinent called Pannotia on the South Pole. Pannotia was formed when a previous supercontinent called Rodinia fractured apart, but Pannotia itself would be pulled apart by tectonic forces not long after it formed. You breathe with an oxygen tank to handle the low oxygen of the Ediacaran. The silence is eerie, and the crashing of waves are the lonely soundtrack. Something comes over you. It's too quiet. You yell as loud as you can, shattering the silence. You have a crazy thought. Was that the loudest sound that ever occurred on Earth in the past year? Or even the past million years? You look towards the land, where there is nothing but red rocks as far as you can see. In the sky, you see a gibbous moon. It looks unnaturally huge and bright. In the Ediacaran, the moon is 19.3 kilometers closer to Earth than our present day. Somewhere on the horizon, a bolt of lightning announces some distant storm, pouring rain over some savage and deserted landscape. Many of these storms would have created massive runoffs and mudslides without tree roots to stabilize riverbanks. The sediment would create temporary rivers and would constantly break and reshape the land. Around 10 million years from this time, in a place that would one day be Australia, erosion runoffs just like this would create the very first rock layers that would one day be sculpted into the natural monuments of Uluru and Katachuta. On land, you are the only thing alive that has more than one cell. But beneath the waves in the shallow tidal zones, the second main Ediacaran assemblage was thriving. You walk down to the water's edge and you squat down to touch the ground. Something about the shallow ocean floor is off. It's not soft and sandy like it's supposed to be. Instead, it's covered with a tough, slimy ooze on top of the sand. It's called a microbial mat, and is made of layers upon layers of bacteria, linked with slimy polymers and sediment particles. These biofilms are built up over millions and possibly billions of years. They are a nutrient-rich layer called a substrate, and while only the very surface is made of living bacterial cells, they can become centimeters thick underneath. Microbial mats create a tough surface and a barrier between the water above and the anoxic sediment below. This mat ground layer was so thick that the microbial mat itself sometimes fossilized, creating a ridged texture sometimes known as elephant skin. You spot something moving very slowly. It's a big, flat oval about the size of a dinner plate, with a line down the middle and segments that pattern out in a ribbed shape. It has bilateral symmetry and appears to have a front and a back. It's an animal called Dickinsonia, and you watch it moving along the shallow seafloor, feeding on the bacteria of the microbial mat. This is an exciting development. It is an animal that can move under its own power, making it one of the most advanced adaptations on Earth at this time. There are lots of other Dickinsonia all around. Some as small as a coin, and some really big ones, 1.5 meters in length in the deeper waters. While Dickinsonia is most likely an animal, it still holds many mysteries. Was this an early worm, an arthropod, or maybe a humongous protist? Did it have any ancestors, or did its lineage end forever in the deep fog of the Ediacaran? You spot a slug-shaped animal called Cambrian, 10 centimeters long, with a dome shape that appears to be a soft, non-mineralized shell. It might be an early ancestor of mollusks, and you watch it sliding along on a single slimy foot that ripples as it moves. It has a proboscis coming out of its head, and as it moves, it scratches into the microbial mat to feed, leaving a trail in its wake. Another grazing animal emerges from a crevice between some early sponges. This one is called Sprigina, named after Reginald Sprigg. It is as long as your finger, with a very defined bilaterally symmetrical body plan. As it slides around, you can clearly see its head and tail, and it looks remarkably like a simplified trilobite. But like Dickinsonia, we don't really know what this thing is. There's a boulder that sticks up out of the water. You look closely at the base, where a cluster of a dozen tiny little animals called Tribrachidium are tucked into the crevices. They are only as big as a button, with a triradial symmetry that creates 3 arm spirals that come from the center. They use these structures to funnel water into their central point and filter feed. A little deeper, past the breaking waves, there are clumps of sessile animals anchored into the microbial mat. They remind you of Charnia, but these were not rangiomorphs with fractal bodies. Instead, they are a different and possibly related group called the Ernietomorphs. One of them, called Ernietta, look like upside-down bells, partially buried by mud. As each wave passes, the thin fronds of the Ernietta wave around and filter nutrients from the water. Lying in between the Ernietta groups, you see a long, segmented animal called a phylozoan lying on the seafloor, but it's difficult to tell whether it's crawling along or if it has an anchor and has fallen over and died. Sharing space with the Ernietta fronds and the phylozoan are strange animals called Funisia, which are attached to the microbial mat and stand up tall like upright worms. They are clumped in colonies about 30 centimeters tall, waving wildly with each current. While paleontologists are unsure whether they are worms, sponges, or cnidarians, these are some of the first multicellular animals to reproduce sexually. All of these weird and mysterious animals are part of a group called the White Sea assemblage, which lived between 560 to 550 million years ago, and have been found in places such as Russia and Australia. By the end of the Ediacaran, animals were innovating new survival strategies that might have contributed to the downfall of the entire Ediacaran biota. Starting around 542 million years ago, burrowing worms called Treptichnus were exploiting a new niche. They were digging tunnels in the background sea floor, eating the tough bacterial mat that provided food and anchorage to so much of the Ediacaran world. As they profited from complete freedom to eat as much as they wanted without competition, they were eroding the background over millions of years until the start of the Cambrian, 538 million years ago. The burrowing had another effect. Treptichnus worms were opening up patches of an aerated, soft, silty ocean floor, like the one you feel when you walk into the water in a river or a beach. As the microbial mat disappeared, rangiomorphs couldn't anchor down, and Dickinsonia couldn't graze. This event, called the Cambrian substrate revolution, or the agronomic revolution, was such an important change that it marks the Cambrian Ediacaran boundary. The last main assemblage of the Ediacaran was the Nama assemblage between 550 and 539 million years ago, based on fossils found in Namibia, Oman, Canada, and Siberia. In this shallow water environment, animals were becoming more mobile as they competed for food. While there were some filter-feeding Ernietomorphs like the leaf-shaped Swatpuntia, other animals were starting to gear up for an arms race. One of these was a 15 centimeter long animal called Cloudina, which developed hard, biomineralized shells that stacked into each other like a tiny string of nested cups. Another defensive animal was the 3 centimeter long Nemocalathus, which attached to the seafloor with a hollow stalk, leading to a mineralized cup shape that was perforated with small windows. If these animals had hard shells, this must mean they needed protection from something. Predators were evolving. Some worm fossils from this period showed evidence of an early digestive system, and some of the first teeth. By the end of the Ediacaran, some burrowing Treptichnus fossils indicate that they changed direction to avoid other animals' burrows. If they detected another burrow, then this might mean that animals were developing sensory organs, maybe even the first eyes. By the turn of the Cambrian, the Ediacaran biota was disappearing, after 20 million years lazing around on the microbial mat. The loss of their microbial mat habitat is a major factor, as well as a global glaciation called the Baconurian glaciation that froze the planet at the end of the Ediacaran. Another factor was the appearance of new and sophisticated predators that were appearing during the Cambrian Explosion that would have seen these soft, defenseless ovals like Dickinsonia as nothing more than a free pizza lying on the bottom of the ocean. For some body plans, like the fractal shapes of Fractofusus, they were an evolutionary dead end. If the creature could only create fractal copies of fronds, it could never evolve new organs like eyes or limbs, and its evolutionary journey was doomed from the start. Cambrian animals were on the way, and the arms race was on. A new world was appearing, one in which animals needed shells, sensory organs, eyes, muscles, or teeth to be a predator or survive from predation. Life was finding a new place in the new ecosystem, and the defenseless soft-bodied Ediacaran fauna, like Dickinsonia or Kimberella, didn't stand a chance. On the shallow beach on the shores of Pannotia, you wade out of the water, trying not to step on the slug-like Kimberella that are sliding around, grazing in peace. Just before you leave the Ediacaran, you take some time to watch a huge Dickinsonia the size of a coffee table drifting along the green slime, enjoying its easy meal. You spot a string of small empty shells, each one the shape of a ring and the size of a button. You pick them up, handling them gently so they hold together. It belongs to an early shelled animal called Sarina. How unique, what a great souvenir to remember the Ediacaran. You watch the waves push at these simple, vulnerable creatures, just kind of chilling out. It seems like a peaceful and innocent time, before the arms race of predators and prey, before swimming, walking, hunting and flying. This is the very last time an organism could survive just by being passive, before life gets truly competitive. If you'd like to know more about the worlds of deep time, make sure to like and subscribe.